Prashant ji, good evening.
Good evening, Jitendra. Good evening.
Good evening, Prashant ji.
Good evening, Jitendra. Good evening.
Okay. Got your voice, sir.
Hello. Good evening.
Good evening, Miss Upasna ji. This side Jitendra Malik, login from Dr. Anupam Sibal sir's account.
Hello, Jitendra. Am I audible clearly?
Yes, ma'am. Loud and clear, ma'am.
Okay, thank you.
ये है presentation. Good evening, sir. प्रशांत बोल रहा हूँ.
हाँ प्रशांत.
हाँ सर, ये presentation मैं upload कर दूंगा.
हम यहाँ से भी presentation दे सकते हैं.
अच्छा सर, मैंने वो second slide जो acknowledgement का था वो last में किया हुआ.
नहीं, हम यहाँ से दे रहे हैं presentation. आ रहा है?
ठीक है.
Okay, ठीक है.
तो इसको बंद कर देता हूँ.
It is better that I control it from
Okay
here.
sir. All right sir. Thank you.
ये कि तुम मूव कर रहे थे प्रशांत?
हाँ सर.
ये presentation आप मूव कर रहे थे?
हाँ सर.
तो करो, फिर आप ही करना.
ठीक है सर.
जब ठीक हो रहा है तो आप ही करना. पूरा स्लाइड शो कर देना जब होगा तो.
Good afternoon, sir.
Good afternoon.
Good evening, sir. This side Upasna Mukherjee.
Upasana, good evening, Upasana.
Good evening, Dr. Gangli. This side Jitendra Malik from Gapio, sir.
Hi, Dr. Malik. Dr. Nand Kumar has joined.
Not yet, sir.
Okay.
Sir, bottom here you can see the slides. Yes, slowly. तो मैंने अभी पॉज किया हुआ है इसको.
हाँ.
अभी पॉज कर दिया शेयरिंग.
क्या बोल?
Prashant,
Okay.
Prashant sir, we can see the slides till now.
All right sir.
They have shared it. Okay.
Sir, stopping.
Hi, Jaswinder.
So the introduction is with you, no? For Dr. Nandakumar and Subhas. Hello. Uh-huh. Achha, achha. Excuse me, sir. प्रोफेसर गांगुली सर.
हाँ बोलो.
सर वो इंट्रोडक्शन आपके पास है ना उनका?
Introduction है. I have to introduce Dr. Nand Kumar and
Yes, sir, yes, sir.
Upasana.
Yes, sir. Yes.
No? Okay.
Thank you.
And I have to also introduce myself.
Yes, sir. In the beginning.
Somebody
Sir.
will introduce me.
Just 2 words about yourself.
And just let me know that.
जितेंद्र जरा फ्लो बता. Mr. Jitendra.
Yes, sir. Sir, Miss Upasana will introduce you for your talk, sir.
Achha, you will introduce me during my talk.
Yes, sir.
Yeah.
So I will introduce Dr. Nand Kumar.
Yes, sir.
And then Upasana.
Yes, sir.
Okay.
And sir, and something about the meeting also. You start with the.
Yeah, I will say a few lines about the meetings.
Dr. Jaswindra, may I request you to check your slides if you want to, sir?
Yes, I am checking. आपको शेयर करनी है क्या?
नहीं, जैसे आप अपना प्रेजेंट कर देना, बट आप देख लो आपका स्लाइड वगैरह ठीक से चल रहा है कि नहीं.
तो शेयर करो सर, शेयर करके देखो.
आप अपने लैपटॉप से शेयर स्क्रीन करके सर करिएगा प्लीज.
अभी कर दे रहा हूँ सर.
जी.
आ गई, शेयर हो गई.
हाँ हो गया,
हाँ
हो
जी
गया.
सर.
इट इज विजिबल सर. दैट्स इट.
अभी चेंज हो पा रही है ना?
बिल्कुल है. PGI Chandigarh, we always start on time and end in time. There is a great opportunity. Actually, when in the GAPIO Executive Committee, I suggested that we have a meeting on omics for decision making for clinical community and other scientists. It was kindly accepted, and today we are for the same purpose. I am Nirmal Ganguly. I was former DG ICMR, at the moment AHF president, and also executive committee of GAPIO member, was director PGI Chandigarh, JIPMA president, etc., etc. And today my co-chair is Dr. Nandakumar Jairam. He chairs the largest hospital chain, Medica Healthcare Group, also president of his current president of the GAPIO. He also was chair of the Columbia Asia Group and is a famous minimal access surgeon. My moderator today is Dr. Upasana Mukherjee, and she is a geneticist. She has a master's degree. She is a certified geneticist, genetic counselor. She works in the Apollo Kolkata and also Navajato Shishu Pratishthan in Calcutta. She is also attached with the Eurofin and has published a lot of papers in the subject and area. So she will be moderating it today. So can I request Dr. Nandkumar to say a few words? Then I will. Before that, I will talk about this particular conference. Today, we have a galaxy of speakers. and they are authorities, and some of them are clinicians and some of them are basic scientists, and but all of them have done tremendous contribution in the area. And it will be a bouquet of the different cancers because we thought that we should have a commonality. So the focus is on cancer and the decision making in cancer, clinical decision making in cancer with whatever data we have. So Dr. Nandkumar, will you please say a few words? Dr. Nandkumar is in a car, so he might have been cut off. So I will start with my presentation and then hand it over to Upasana. So if you could show my first slide.
I think Ms. Upasana will give a brief introduction
Okay.
of yourself.
Please come on, Upasana.
Good evening, sir. Thank you so much, first of all, for giving me this platform and for the wonderful and humble introduction. Good evening, everyone. Today's topic is very important and interesting to among all the geneticists and the researchers, even the clinicians in India and I believe across the globe. Today's discussion will be on it is on the genomics, and we will be talking about the multiple omics researches, multiple omics, how the omics studies, like from disease discovery to the precision medicine, how it is helping us and will be taking us towards the better future when we will be actually knowing about the precision medicine and will be targeting each and every genetic variant. This topic will definitely enlighten us towards our goal. To start with this, our first speaker, Professor none other than Professor N.K. Ganguly. He is former president of National Academy of Medical Sciences, Indian Science Congress, and JIPMAR Puducherry. Professor Ganguly was former Director General of the Indian Council of Medical Research, also served as the Director of PGIPMAR, Chandigarh, and National Institute of Biologicals. He has been conferred with 6 Doctorate of Science degrees by the Indian universities. He has guided or co-guided more than 158 students for their PhD theses and authored and contributed to more than 800 academic publications and 21 book chapters. Professor Ganguly has received 9 international and more than 114 national awards. For his immensely valuable contribution to the medical science and exceptionally distinguished service, Professor Ganguly was awarded the prestigious Padma Bhushan Award by the President of India in 2008. We welcome you, sir, for the kind session, the next session on use of multiomics in determining responsiveness to the lung cancer. Welcome, sir.
Thank you, Upasana. If you could come to the first slide, please. Okay, so in this slide, you see the power of the checkpoint inhibitors. You see a lady with a fungating oral cancer, head and neck cancer. And after the treatment, you see that how the cancer has disappeared. So, but today I will be showing you the data of the NSLCs, that also squamous cell cancers, because here you could give, because of the US FDA, the checkpoint inhibitor, next slide please, inhibitor, alone without any other adjunct therapy. So checkpoint, normally the cancers have an immune evasion. There are 2 immunologic signals. One immunologic signal is done through the T receptor cells, is the primary signal, but the immune response actually is completed when the secondary response to the co-stimulators happen. What cancers do, they actually mimic with molecule which resemble the co-stimulating molecules, and they develop a ligand. So they're known as PD-1 and PDL ligand. And when this ligand works, it blocks all the immune responses. So the immune responses, which are the major protector in the cancer, they really don't work during the most cancers. So recently, about 6 cancers, these checkpoint inhibitors, the major developer with others is Vijay Kachuru, who is in the Harvard, and they actually brought in this many of the protocols and which are used at the moment for cancer treatment. 6 cancers, you can give primary checkpoint inhibitors. Next, please. So, we have lot of. There was an acknowledgement slide. I don't know where it disappeared. There are about 6 people who have contributed over here. I want to show their slides. This work is Kriti Jain, who is my PhD student. For the immunology work, Dr. Amit Awasthi, who comes from Harvard, was a partner. The Shyam Agarwal was a medical oncologist. Rashmi Rana was the proteomics person. And we had Deepak Rathore and Bhavtosh Das for microbiomes. And for the inflammatory response, we had Surajit Ganguly. And our statistician was Payal Chugh. Now show me go back to the slide here. So we had this is our protocol. We from the PBMCs, we have done all the immune responses. We have around 5 panels. We designed 5 panels with 8 different reagents and looked at most of the responses of the adaptive as well as the innate immune systems. We took plasma and did proteomics into this, as well as metabolomic with the plasma cells. And we also did with the serum, we did the cytokine, and we used the Luminex system for that. We did an NGS also with the tissue samples which we got, and we also did microbiome, and this was also done through sequencing with the Bhavatosh's lab. Next, please. And for the metabolomes, we were helped with Dr. Jassi. So these are the results which I can tell you, because we don't have time to show you the detailed results. So we saw that plasmacytoid dendritic cells were up in the responders, because the main question, we asked only one question, because only 30% responded to checkpoint inhibitors. We asked the question, and they're frightfully expensive. So in 70%, they don't work, and people lose their home and hearth because of these expenses. So we asked a question that whether this person will respond or not respond, and while the person is being treated, the person is responding. So we wanted to develop through multi-omics platform a complex set of biomarkers. So one of the major and if it was a 4-fold increase, only then from the control, only then we called it significant. So the dendritic cells, plasmacytoid dendritic cells were high in these responders, and myelocytoid dendritic cells were not high; they were low in the responders. The NK cell, the NK T cells were enhanced in the responders, and there are 4 populations, but one of the NKT cell, iNKT cell, was enhanced. Then the new antigen specific lymphocytes, which are the CD8 plus and PD1 plus, they target tumor specific new antigens. This was significantly increased in the responders. The granzyme B, which is indicating for the increased cytotoxic potential of T cells, was significantly increased in these. and the CD8 cell enzyme in the immunologic system. We couldn't differentiate the subspecies of the enzyme, but with the proteomics, we could know that this was enzyme B or enzyme 2. Inflammatory monocytes, possibly indicative of tumor metastasis, was also increase in the non-responders, because this particular thing indicates metastasis. The major CD4, CD8 cells which had Ki57, that is the activation markers, were increased in particularly the CD8 cells, which target the tumor cells if we can make them free of this. The cytokines, interestingly, the 3 cytokines were targeted. After we did about 40, but 15 were done, 16 were done in the Luminex quantitation. Proteomics, we found 60. So the IL-21 and IL-23 were found high in the responders, and pathogenic IL-17 was also high. So IL-23, IL-21 actually move it towards from PI3K to through mTOR pathway to AKT. AKT, you know, is responsible for tumor proliferation, angiogenesis, etc. So it dampens the AKT. So this was our immunology, immunomes. Next, please. Next, please. So in the genomics, we didn't find much in the TMB because the tumor mutation burden, microsatellite instability, and PDL, these 3 are normally used at the moment. The microsatellite instability was not useful, but we found 2 things. We found that desmocholine 3 secretory PDL1 was high, and desmocholine 3 was also high. There were 2 other mutations which we found. TP53 gene mutation, which is useful for breast cancer and others also for tackling the tumor, and FR4P was also. There's a very strong mediator for taking care of the immunosuppression. This was also mutated positively in our genomic system. There are a huge number of signatures. I'm only talking of the significant signatures. Next, please. So I will talk a little bit of desmocholine 3. I want to work with Dr. Sudeep on this. And this is an addition molecule. This also suppresses the AKT pathway. This we have used for biomarker for differentiating squamous cell NSCLC from adenocarcinoma. Next, please. Next. So this is one of the overall survival data from a Chinese group, and we can see that those who have desmocholine 3, they respond favorably. Next, please. This is the treatment with Mycidec, which is already in. This is an immunomodulator, multi-toll receptor modulator, already is in the market for the squamous cell NSCLCs, and there's a 16-week data, Kaplan-Meier, and you can see that there is a definite advantage if there was desmocholine 3 positive cells. So this shows that the genomic data is a demonstration that genomic data you could use for the personalized medicine also. Next, please. So this is the desmocholine 3 result in our group, and you could see that it actually expresses very high in the responders compared to non-responders. Next, please. So this is the metabolome data. This here we find that sphingosine pathway, which leads to the ultimately ceramide pathway, is not good for the responders, and we now know the reason. That Dr. Blanket, who died recently, found out that the NKT cell driver is the ceramide, And so the NKT cells are one of the major protective mechanisms in the innate immunity in cancer. And if they are dampened by the sphingosine, then ceramide, then it becomes a problem. The hydroxybutanone, gentisic acid, and glutathione all were bad for responders. So, but arachidonic acid and glutamate metabolism was beneficial. Glutamate metabolism ultimately helps in the leading out from the tumor suppression, as well as better response of the system. And arachidonic acid, which is one of the inflammatory cyclooxygenase pathway, ultimately helps in the penetration of the immunologic cells into the tumor substance when it is, particularly when it is very dense. Next, please. So these are the Kaplan-Meier for the urea, and we can see that 5-year survival. This is very good, whether you look at the AUC and whether you look at the responses. Next, please. So this is the key results of the microbiome, and we see that bifidobacterium. This was done through the MySec analysis, and Bhavtosh helped us in this. We can see that the bifidobacterium, Doria, as well as one of the and from Polyphemonas, and one of the numbered microbiome were high in the responders, and the Klebsiella as well as the Enterobacter were the baddies, and they were high in the non-responders. This was a very significant data. Next, please. And when we correlated it with the metabolic pathway, it correlated very well. Next, please. So what we are doing now, and in the targeted proteomics, which was done with the NovaSeq and Olink linkages, we found that they were moving very well in the responders and non-responders. The ones which moved in the non-responders were IL-13. It is one of the inflammatory pathway. Ganzyme A, VGFR2, tweak, Ganzyme H, and CD27 moved in the non-responders, and Ganzyme B, CRTM, and CCL23, which is in the apoptosis pathway, moved very well in the non-responders. Next, please. In the responders. So this is how it is moving. I will not show you the details because it will take a lot of time to show the individual patients here. Each spot is an individual patient. Next, please. Next. So this is a summary, and you could see that in the responders, increase the T lymphocytes, the plasmacytoid dendrocytes, activated T cells, proliferated T cells, monocytes, NK, Ganzyme B expression, and baseline CD8 was increased. We've also got secreted PDL1, we got IL-2, IL-23, as well as IL-21 increased in the responders. In the tumor, there was decrease in desmocollin 3 and PDL1, and Ganzyme A, VGF2, tweak, and Ganzyme H and CD24 decreased in the responders, in the non-responders. And in the responders, we found TMB, TP53, FGFR4 as well, so in the genomic signature, and CCL23, CRTM, and Ganzyme B in the proteomic signature. In the metabolic signature, urea, arachidonic acid, and glutamate were increased in the responders, and sphingolysine, ceramide, butanone, glutathione, gentisic acid, sphingolipid metabolism pathway were responsible for non-responders, and Enterobacter Klebsiella were baddies, and there was a xenobiotic degradation pathway in the non-responders, Bifidobacter, Doria, Polyphemonas, and FEM family and lysine birth-in pathway where enhanced in the responders. So we can do something. We can enhance the lysine biosynthesis pathway by providing appropriate, and here also we have designed an intervention. That intervention is the fecal transplantation. We have done about 17 fecal transplantation cases in these, and we hope that those who were partial, we are using RECIST criteria. So those who have responders are quite well, but those who have not responded but are partial responders, we want to make them responders by giving them a fecal transplantation. Next please. So this is the slide that at this given moment, the multi-omic approach could actually help the patients in selecting that who are responders and treating them ethically and with equity. And also, though we can use some of these for additional therapies to checkpoint inhibitors to help these patients. Thank you very much for your attention. And this is a cured subject. So we are doing in other cancers also, but today it was lung cancer. And I'm very happy to tell you that around 50% of our selected patients have survived for 5 years and more in the lung cancer. Thank you. Upasana, back to you.
Thank you so much, sir. Thank you so much for this insightful talk. I would request the audience to post your questions on the chat box. We'll be taking the question answer session at the last after all the talks given. Not wasting a little more time, moving to the next topic, I would like to now call Dr. Rashmi.
Yeah.
1 second, sir. Yes. I would like to call upon Dr. Rashmi Rana for the topic, for the next topic. Dr. Rashmi is a highly accomplished professional in the field of biomedical research and serves as a consultant in the Department of Biology, Biotechnology and Research while holding the position of Associate Professor in the GRIPMAR within the prestigious Sir Gangaram Hospital in Delhi, India. Dr. Rana is a passionate and prolific researcher with a remarkable portfolio of over 20 published papers in renowned indexed national and international peer-reviewed journals. She is also an editor of various journals such as Frontiers in Molecular Biosciences and British Journal of Cancer Reports. Dr. Rana has earned recognition through various national and international awards and fellowships, such as the prestigious ESMO, ASCO, Bill and Melinda Gates Foundation Fellowship, the HCFI Dr. KK Agarwal Research Fund Award, Biha Award in the Clinical Sciences, and the Young Scientist Award. Welcome, Dr. Rana. Over to you for your next topic on multi-omics in the glioma.
Thank you for the nice introduction. So very good evening to all. So myself Rashmi, today title of my talk is the multi-omics in glioma biology. So can you hear me? Can you?
Yes, Dr. Rana. Yes.
Okay. So I'm talking about today I'm talking about the brain tumor, that is of glial cells consists approximately 40 to 55% of intracranial brain tumor. So as per the WHO criteria 2021, they includes astrocytoma, oligodendroglioma, epididyma, and mixed glioma. So this is the right side showing the overview of the glioma. As you know, the prognosis of because of the size, location of the tumor, psychological and physiological pattern, and malignancy grades of the tumor. So till date, there is only few treatments are available of glioma: surgical incision, that is craniotomy, chemotherapy in combination of radiotherapy. For the diagnosis, there are few diagnosis methods are available: physical examination, neurological examination, scan of brain, MRI, and CT scan. So the clinical manifestation because of this glioma is headache, hemiparesis, depression, aggression, the vocal and visual difficulties. So today's problem is the basically which we are going to encounter is like problems is that the overall survival of the glioma patient is about 6 to 15 months with poor aspects of life, and till date there is no treatment available to distinguish between the grades without surgical interventions, as well as the proper treatment is not available which can increase the survival rate with better lifestyle. So here we are taking three-dimensional approach of our team. Number one, galectin-3 based diagnostic and therapeutics. Two is the CRISPR-based approach in therapeutics, and third one is the exosome-based drug delivery. So today my focus on only first one, that galectin-3 based diagnostic and therapeutics. So today's talk, there are three components which are extensively involved. Number one is the brain tumor. Number two is the extracellular vesicles, and the three one is the proteomics. That in one approach is mass spectrometry. So I'm just giving you the glimpse of the EVs. So what is EVs? EVs is a extracellular vesicles. Their work is basically cell-to-cell communication, as well as they carry the information between DNA, RNA, and protein to another cells. So as per the literature, they're showing their role is the tumor diagnostic, prognostic, or drug nanovehicle. So we have published couple of articles in this regard. So there are three components which are involved in today's talk. So the description of the work. So we have been collected blood and blood as well as tissue samples of different grades of glioma and healthy individual. So this protocol is already approved from Institution Ethical Committee, Sir Gangaram Hospital. So our inclusion criteria is subjects with 18 years or above, and they are histologically confirmed glioma as per WHO 2021 guidelines, and willing to give consent were be recruited for this study. Subjects presenting non-tumor condition of the brain, head injury, and willing to give consent were considered as a healthy individual. For exclusion criteria, subjects under 18 years age and unwilling to give consent were excluded from the study. So this is the methodology. We isolated extracellular vesicles of different grades of glioma through ultracentrifugation methods. I'm not going to the much details. And finally, we also published this method in Current Trends in Chemistry 2021. So though we isolated EVs, as I am earlier talking about, from different grades of glioma. So whether those are EVs and any other, extra vesicles, so we have to confirm and characterize with the different technology. So here we use flow cytometry, nano tracking analysis, as well as Western blot. So these are the result. Here you can see the CD9, CD63, and CD81. These are the exosomal biomarker. These are basically tetraspanids that show the enrichment of the extracellular vesicles in the fraction. So here you can see the population of CD9 is showing a very promising, and CD63 as well as the CD81. Also we check the size with the help of nano tracking analysis. The size of the EVs is 30 nanometer to 180. So here we found 35 actually. And also the Western blot is showing control samples, EVs, grade 1 and grade 2. Here you can see the expression of this CD63 increase and grades increased. So after characterization, we confirm that whatever we are isolating, those are the extracellular vesicles. After what we did, we trypsin digestion those extracellular vesicles exosomal proteins with the help of trypsin, and finally we run on the mass spec. After mass spec run, we did in silico analysis, and we verify with the help of very specific and sensitive technologies, ELISA. So these are our proteome and bioinformatics analysis. Here you can see this image A is a vesiclepedia. Vesiclepedia is a Indian bank, is like a PDV bank. So here 8,353 is proteins, glioma proteins is already available, and 99 were common we have found in our result. 24 is novel protein which we have found in our different grades of glioma in comparison to control sample. Here you can see in the C diagram, this is the PLSDA analysis of replicates of the run of the samples. Also, gene ontology, figure A showing the gene ontology pathway analysis, and that were found to be enrichment of after functional annotation of identify using this biological process, cellular component, and molecular function. Also here you can see we found two pathways which are the involved in the progression of the glioma. As disease severity is increased, this pathway also we found in very high, in this grade 1, grade 4. So here you can see this a cholesterol metabolism. So there was earlier study showing it was published in the PNAS that also supporting our data that we also found that cholesterol metabolism is participating in the progression of the glioma. So this Venn diagram indicating the common differentially expressed proteins between the stages. Also, we found the 7 common one. In that 7, those 7 one are the, twofold chain. As more as disease condition increase, the level of this particular proteins also increase. After that, after many filters and using bioinformatics, we found there is a one protein biomarker that is galectin-3 binding protein. Here you can see the interactome protein and their fold chains between the different grades of glioma in comparison to controls, and the blue line represent the direct interaction between galectin-3 binding protein. So here, once we found that galectin-3 having any role in the progression as well as it can be used as an early detection biomarker for glioma. So we validate and verified in larger cohort of the samples of glioma grades in comparison to healthy individual. So this left side is the bar gram showing the plasma sample. So here we found the concentration nanogram per ml of protein, and that was statistically very significant if you see the comparison between the control versus grades. Also same we found in the extracellular vesicles proteins. That means we verified in the vesicles also along with the crude samples. Ultimate we have to check in the plasma only. So based on that, we summarize here that galectin, this work is already published. We already mentioned we have isolate and characterize. After that we did LCMS, functional analysis take place, and verification already done. So after that we thought we also verify in the tissue sample of the different grades of the glioma samples. So in tissue sample we perform immunohistochemistry using our methods, histopathological methods. Here you can see our graph. If you see this, the photograph, that control, this is the control image, and we are using as a primary antibody is a galectin-3 binding protein. Ultimate we have to prove in our tissue or tissue. So grade 1 you see the expression of this, and this is the grades 2, and as the grades increase, the level or expression of this galectin-3 protein is increased. We have analyzed in 40 samples of each grades of glioma. Also, we found one image that is the mixed glioma basically. In mixed glioma, oligodendro, astro are involved cells. Also, we found that if you see the expression is very, very high. So we perform this immunohistochemistry analysis in different dates of glioma. Here you can see the same pattern which we have found ELISA as well as bioinformatics analysis. So it's very statistically very significant. After that, we also thought why not we perform flow cytometry. So we also check their expression with the help of flow cytometry. Method in so here you can see the unstained, the population parent 1.5. This is the unstained slides. This is the healthy control. See the expression of the and the grade 1, grade 2, grade 3, and grade 4 as the the increase manner which we have found earlier. So we also proved also with the help of flow cytometry, and it got statistically significant. After that, also we thought, can we do immunocytochemical analysis with the help of this GAL3BP? So we also found the same thing, like we are pattern showing. Here you see is a 10x. Elexaflor we use as well as DAPI, and our primary antibody obviously galectin-3 binding protein. So here you can see between grade 1 and grade 2. Also, grade 3 as well as grade 4. Expression is increasing as grades increase. Also, we calculated, and this showing the representation of GAL3BP positive cell. We perform in 11 samples yet, and we still found statistically significant. After that, this verification, validation, we thought, can we use that GAL3BP as an early detection biomarker using blood plasma samples of glioma patients? Yes. So what we did in that, we developed a GAL3BP detection lateral flow assay initially. So this is the procedure which we are going to, how we are going to, develop this lateral flow assay. So this is just a protocol. And we also, patented this work through Government of India. And we are also going to do MOU with Oscar Biotechnology Private Limited to develop, early detection methods. And also simultaneously, we thought, can this protein having the role in the progression of the glioma, also having as a diagnostic early detection marker? So could that protein is having any therapeutic value? So we thought that, and we found a very prominent result with the help of molecular docking. This particular one is the collaboration with All India Institute of Medical Science, Department of Biophysics. One of my colleagues, Pradeep Sharma, is in that. He performed the experiment of molecular docking, and he found there are 3 inhibitors. You see in the table, there is the values very, very significant. And what we did, we performed and we submitted in the British Journal of Cancer Reports. This is under review right now. So these are my projects which are ongoing projects, my infectious disease as well as oncology and other projects. These are my awards, and these are some publications. And I acknowledge definitely Professor N.K. Ganguly, my mentor, my guidance. Because of there, I'm here, or my team, my research scholars, as well as clinician, patients, and my collaborators. So thank you so much for your kind attention and time. This is my team. Thank you. Hello.
Thank you so much, Dr. Rana. This was wonderful, and really I was amazed to see the kind of extensive study you have done for the protein. This is wonderful, commendable. Thank you so much for your time. Moving to our next topic now, we will be taking the questions. Audiences, I request you to post your questions on the chat box. We'll be answering and addressing them at the last of the sessions. Moving to our next topic, which is on use of the omics in the clinical settings of cancer. It will be delivered by Dr. Sudeep Gupta. Dr. Gupta is a professor of medical oncology, director of ACTRATE at Tata Memorial Care Center, Mumbai. He is an internationally renowned breast and gynecological cancer oncologist. He is a prolific clinical researcher who is actively involved in planning, organizing, conducting, analyzing the phase 1, 2, and 3 clinical trials, and has been the principal investigator or co-investigator for more than 120 trials. Dr. Gupta has published more than 250 papers in the peer-reviewed journals. Without wasting a single more time, welcome you, sir. Over to you.
Hello, very good evening, good morning. Am I heard?
Yes, sir.
Yes.
Thank you very much. I would like to thank Professor Ganguly for the opportunity to speak at this excellent conference. To the left is Tata Memorial Hospital, to the right is ACTRATE in Mumbai. And what I will do today is to talk a little bit about the use of omics in the clinical settings in cancer. Several years ago, we audited a single year's data of breast cancer patients presenting to Tata Memorial Center, and we found that about 30% of our patients had the triple negative breast cancer phenotype. This is in contrast to only 10 to 15% such patients who are seen in Western data. And the reason why this is important is because triple-negative breast cancer is a more aggressive variety of breast cancer with a higher risk of relapse in the initial years after diagnosis. Last year in December, I had the privilege of presenting a randomized control trial at the San Antonio Breast Cancer Symposium as an oral presentation on behalf of the Breast Cancer Working Group. This randomized trial looked at the addition of platinum to standard taxane anthracycline chemotherapy in patients with triple-negative breast cancer who had non-metastatic disease. We accrued in this trial for 10 years a total of 720 patients, of whom 717 were eligible for analysis. And we reported the data at a median follow-up of about 67 months. The reason why this study is important and has been hailed as a landmark all over the world by breast cancer doctors and patients is because this is the very first proof that platinum actually improves outcome in patients with non-metastatic triple-negative breast cancer. So you can see that the addition of platinum resulted in a 7.5% increase in overall survival, which was statistically significant and also highly clinically meaningful. When we broke down the data by age, we found that younger patients had a huge benefit of about 11% increase in overall survival from 65% to 77%, which was highly statistically significant, but older patients had no benefit. So we concluded that carboplatin should be considered as a standard of care in patients with triple-negative breast cancer who are either younger or premenopausal. We also concluded, and I have not shown you the data today for paucity of time, that increased pathological complete response is highly predictive of survival outcomes. So we were interested contemporaneously with the clinical study in understanding the clonal evolution of tumor in TNBC patients. And we hypothesized that tumors are polyclonal from very early in their natural history. So what we did was to collect longitudinal samples from individual patients. So each patient's tumor sample and blood sample from the time of diagnosis in the non-metastatic stage to first relapse and to subsequent relapses right through to their deaths. And we collected this sample set from 7 patients, of whom from 3 patients there was enough sample for analysis at all time points. And you can see that we subjected them to multi-omics, high-depth exome sequencing at 200x, SNP arrays for copy number aberrations, paired end RNA sequencing with 50 to 100 million reads per sample, and then a targeted resequencing of both the tumor as well as the circulating tumor DNA at more than 20,000x. So we constructed the life history of triple-negative breast cancer tumors. We found that the mutational burden varies over time, is generally lower in non-metastatic stages and increases as the tumors relapse over time, but that the mutational signature is retained with signature 3 persisting in all tumors in all samples from all patients at all time points. The upper panel in each of these 3 data sets belongs to the somatic tumor. The lower panel, which is outlined in red, belongs to the circulating tumor DNA, and the individual bars are the mutations. And you can see that the mutational profile that is captured from circulating tumor DNA presents a more complete picture of the tumor compared with somatic tissue. And this is at each time point as the patient has disease progression over time. And the reason, of course, is that you can only sample one location of somatic tumor, but the blood will perhaps contain the DNA that is shed from all locations of the tumor. We constructed the mutational clonal evolution of the tumor. And you can see that there is a stem clone, which is depicted as A in every single tumor that persists throughout the life history of this tumor. And there is also the emergence and extinction of daughter clones or subclones as the patient receives successive lines of treatment. And 2 of the 3 patients had a germline BRCA mutation. And this pattern of clonal evolution, which is called the branching evolution, was seen in both germline BRCA mutated and in wild type tumors. And this is the very first demonstration of the branching pattern of evolution in TNBC tumors in individual patients over time. So we concluded, and this is the work of my PhD student Nilesh Gardi, that there is a retention of signature 3 and signature 13, which is the APOVEC signature in all samples from all patients. The copy number profile, which I have not shown you today, is there is widespread copy number change right from the beginning of the tumor's life history, but there is minimal change over time. This is consistent with the previously reported punctuated copy number evolution model for TNBC. And ultra-deep sequencing shows that tumors are mutationally polyclonal with a stem clone that persists throughout its life history, but with emergence and extension of daughter clones. This is consistent with the branching pattern of mutational evolution. ctDNA was very useful and was able to capture mutations not detected in tissue samples. And mutations from the founder clone were expressed at high levels. This was from the high-depth transcriptome that we did. And these mutations perhaps present a very valid target in this hard-to-treat tumor. So, changing tack, I just wish to share with you a little bit about another thought of ours. We know that surgery is the most important treatment for many cancers, perhaps for all cancers. And the way surgery is conceptualized is just as a physical act, but we fail to often appreciate that surgery also induces physiological changes in the tumor, such as hypoxia, wound healing, exposure to anesthetic agents, low blood glucose, etc. And all of these are stressors that act on the tumor. The molecular effect of these stressors on the tumor as it is being operated have not yet been systematically characterized. And we set out to do this in a few studies in the past few years. So what we did was to sample a patient's tumor at the beginning of surgery when it is fully vascularized, in the middle of surgery when it is approximately half devascularized, and at the end of surgery when the tumor is out of the body, when it is fully anoxic. And we subjected these samples to paired-end RNA sequencing, again at high depth, in the discovery cohort of 46 patients. So each of these 46 patients had 3 samples drawn beginning of surgery, middle of surgery, and at the end of surgery. And then we also had a validation cohort where what we discovered in RNA-seq was validated by custom nanostring encounter assay in the 48 patients. So in RNA-seq, we found that there were 737 uniquely deregulated genes in at least one of the 3 comparisons, A versus B, A versus C, or B versus C. We found that the AP1 transcription factor network genes were the most highly deregulated genes in all comparisons. And there was also a highly significant deregulation of genes involved in inflammation and extracellular matrix degradation. And I'm not going into the details because of paucity of time. We repeated the same experiment in patients with head and neck tumors. We sampled the tumor in the beginning of surgery, in the middle of surgery, and at the end of surgery. And we again compared what happens as the surgery progresses. And you can see that again, the AP1 transcription factor network genes are the top deregulated genes in head and neck cancer. We compared the tumor biopsy from a fully vascularized tumor with a sample from the surgical specimen in 7 cancer types in the TCGA dataset. The cancers are mentioned there. So the biopsy sample corresponds to our A sample, and the surgical specimen sample corresponds to our C sample. But of course, TCGA has not done the systematic study that we did. And what we found again, that in the TCGA analysis in 5 of the 7 cancers that we studied, the AP1 transcription factor network was a top deregulated gene network. So we found that the same genes belonging to the AP1 inflammation as well as ECM were highly deregulated in multiple experiments: breast normal tissue, the data of which I did not show you, breast tumor tissue, head and neck normal tissue, head and neck tumor tissue, and in 5 tumor types from the TCGA. We validated these findings by QRT-PCR in 9 patients, again triplet tumors. Again, you can see that the same genes become highly deregulated. And in the nanostring encounter assay, again, the same genes become highly deregulated along with a couple of immune checkpoint genes. So our findings suggest that there is a recurrent pattern, a systematic pattern of deregulation of several pathways that may confer survival advantage to cancer cells if they were to disseminate during the process of surgery. These belong to inflammation, EMT, hypoxia, immune surveillance escape. The AP1 transcription factor network is the consistently deregulated during surgery. This is organ agnostic, tissue agnostic, and this has deep implications for tumor biology and how tumor tissue-based molecular studies are carried out. But you might say that these are just esoteric findings and what is the clinical implication? We believe that what we do during surgery affects the long-term rates of relapse and death in these patients, and can the events at the time of surgery be modulated to improve the long-term outcomes? We performed a randomized control trial of the peritumoral infiltration of local anesthesia. So just prior to the resection of the breast tumor, the surgeon infiltrates a wall of lidocaine all around the tumor. And you can see this a few weeks ago, this was published in the Journal of Clinical Oncology. 1,600 patients randomized half and half into receiving lidocaine all around the tumor prior to its resection versus not. And all these patients are under general anesthesia. So lidocaine was not for purposes of local anesthesia. It was for modulating the stressors on tumors during surgery. You can see on the left is disease-free survival, on the right is overall survival. In 1,600 patients, highly statistically significant and clinically meaningful improvement in both disease-free and overall survival. And on the bottom panel, to the left is local recurrence, to the right is distant recurrence. And both of them are again substantially reduced with the simple act of infiltrating local anesthesia all around the tumor. This was the editorial that accompanied our original article in JCO. And the editorialists from Dana-Farber Cancer Centers agreed with us that it seems reasonable to introduce peritumoral infiltration of lidocaine as a valid measure to improve outcomes in early stage breast cancer. So I have talked thus far about somatic genomics. What about germline? Last year, we published, or a couple of years ago, we published a multi-center study from about 10 centers from all over India to look at the prevalence of BRCA1 and BRCA2 mutations among patients with ovarian cancer. And you know that these are 2 genes that are associated with very high lifetime incidence of breast and ovarian cancer in their carriers. You can see that about 85% of our patients had no family history of breast or ovarian cancer, but we were surprised to find, now the panel that has been highlighted just now is patients with a family history of breast or ovarian cancer, and you can see 55% of them, 55.6%, have a pathogenic mutation in BRCA1 or BRCA2 in their germline. But very surprisingly, even in patients without a family history, and these were 203 patients, there were 20% who had a germline pathogenic mutation in BRCA1 or BRCA2. So a negative family history does not rule out the presence of a germline pathogenic mutation in one of these highly penetrant genes, and therefore it has become the standard of care in India to perform this testing if it is available. We also found that the presence of germline pathogenic mutation was highly correlated with serous and endometrioid histology, but there was no pathogenic mutation in the germline in patients who had clear cell or mucinous types of ovarian cancer. So I've given you a brief snapshot of what we have done. There are a few other studies, but there is not enough time for me to share with you. Let me share with you my final thoughts. Dobzhansky said about 50 years ago that nothing in biology makes sense except in the light of evolution. And over a period of time, I have felt the force of this statement. Where were we in the past? And will this understanding of the past help us gaze meaningfully into the future, at our trajectory to the future? So this is a cartoon that I drew for myself a couple of years ago. On the y-axis is progress, whichever way you define it, and on the x-axis is time. So progress can be linear over time, it can be exponential, it can begin to plateau. And where we will land 10, 15, 20, or 50 years from now, of course, depends on which trajectory we think we are following at this point of time, but also very importantly, some conceptualization of where we are on the arrow of time at the current time point. So these are several collaborators from many different institutions and my own institution, and I am grateful to all of them. These are all the clinicians who comprise the breast cancer working group of Tata Memorial Center. These are my lab members, and I thank you very much for your attention.
Thank you so much, sir. Thank you for this wonderful, insightful, and very, very important topic and the lecture. We shall take the questions at the last. Now, moving to our next very interesting topic, I would like to invite Dr. Jaswinder Singh Maris for his talk on omics in NF, LD, and NASH. Dr. Jaswinder is an assistant professor in molecular and cellular medicine at Institute of Liver and Biliary Science, Delhi. Dr. Jaswinder is the principal investigator and a co-investigator in various research trials. He has published over 34 papers in various peer-reviewed journals and has one patent in his name. He has been conferred with the various awards, including the prestigious Inspire Faculty Award from the Department of Science and Technologies, Government of India. Welcome, sir. Over to you.
Good evening, good afternoon, everybody. First of all, I'd like to thank Professor Ganguly and Professor Nand Kumar for giving me an opportunity. We at the ILBS have started an omics department lately in 4-5 years, and we are doing multi-omics research, and I'm taking care of the laboratory of proteomics and metabolomics. I'm an assistant professor at the Department of Molecular and Cellular Medicine. This is known to everybody. So omics, if I can define omics, omics is the study of DNA, RNA, proteins, and nucleotide in a high throughput fashion. So everybody knows about omics and is looking at the data from Dr. Ganguly and Dr. Sudeep. It does not require much of the understanding here. So you can use different tissues such as brain, pancreas, blood, urine, joints, and you can do multi-omics analysis, which includes DNA, analysis of RNA, proteins, and other things. The idea is to integrate these molecular analysis so that you can identify the molecular map. You can do patient stratification, you can do cell type characterization. Are there certain molecular markers which are there? And can we identify certain drug targets which are important? So the integrative approach lies on to the understanding of the biology per se. So if you see the mass spec based workflow for omics analysis, it includes the sample extraction or collection of the sample, the separation of the sample on using an HPLC, the detection of the molecules that you are going to study, and the data that is required are analyzed. That is the data extraction. And if you see the strategy for biomarker identification, generally we compare a disease with a normal control. We do proteomics or multi-omics analysis to identify a candidate gene. In this case, we can have a candidate protein, metabolite, and then we usually do validation, which is generally a different technique. And this is then applicable on the clinical side. For example, it is used for diagnosis, prognosis, or therapeutic approach. Let me define NAFLD and NASH for you. NAFLD is actually non-alcoholic fatty liver disease. It has excessive of hepatic fat accumulation with insulin resistance. The steatosis is greater than 5% in the hepatocyte. The exclusion of secondary criteria like alcohol-associated fatty liver disease is excluded. So if you have pure steatosis and mild lobular inflammation, this is characterized as NAFLD. And if you have NASH, that is, it is associated with inflammation, it is called as NASH. And NASH has early fibrosis, grade 1, grade 1 is early fibrosis, fibrotic grade 2, grade 3, and then it takes forward to fibrosis grade 4, which is cirrhosis, which then forwards to HCC, development of HCC. That means that fibrosis is the interim between NASH and HCC development. So it is very important to understand what fibrosis is in this scenario. So if you see what is fibrosis, fibrosis is basically generation in any kind of hepatic insult that is happening. It may be NASH, ASH, hemochromatosis, DD, Wilson disease, whatever. If there is an hepatic injury, it leads to activation of HSC. And hepatocyte active HSC is stellate cell activation leads to fibrosis, which further processes to cirrhosis. This time maybe in 20 years, and then it further leads to cancer development. So what are the cells that are involved in cancer development? These are the cells which are involved. The circulating fibroblasts, portal fibroblasts, all gives rise to activation of HSC. And this activation HSC, that is the myofibroblast, is actually responsible for fibrogenesis. So what happens is that if you have any kind of infection like HBV, HCV, you have alcohol, or you have cryptogenic cirrhosis, or any drug or obesity associated, that is NAFLD, that in this case. And if you have inflammation, it leads to activation of HSC, and then therefore there is an activation of myofibroblast, which increases the collagen, it increases the elastin, glycoprotein, proteoglycans, and hyaluronidase. This finally leads from fibrosis to cirrhosis, and then there is development of primary cancer, that is the liver cancer. So what are the works, some of the works on biomarker, as we are working on multi-omics? So these are some of the proteins and metabolites that are identified and are biomarkers for NAFLD. For example, downregulation of ADP, increase of TNF-alpha, decrease of leptin, CRP, IL-6, and oxidized LDL are indicators of NAFLD. If you have NASH, that means TNF-alpha and IL-6 should be increased, and fibrosis, you have increase of leptin. These are known. There are many studies, and I have taken one from this very important study, which has laid down all the known biomarker panels for NASH and FLD. So these are the APRI score, which amalgamates the clinical factors as well as the proteomic studies somewhere where they have used proteins like alpha-2 microglobulin, hyaluronic acid, and they have coined the biomarkers or scores. These are HEPA scores, the fibronic index, the FIBC3 index. So all these index are known, and they have people have studied in number of patients like 500 patients, 1,200 patients, which gives us high AUROC value, and these are the cutoffs. So these biomarkers are already known for NAFLD and NASH, and people are still using this in the clinics. So this is another combination of biomarker where collagen, procalcin, pro-C3, cytokeratin 18, M30 fragment, AST/ALT, these all fragments they have validated in 374 NAFLD patient, which gives us an AUROC of 87%, and this is the sensitivity specificity. So these biomarkers are very well known. So why are we doing more multi-omic studies? Is actually to see the integration of the proteins and metabolites and lipid molecules, and where it leads us to identification of more specific biomarkers. Let's go forward. So I'm present some of the data from our center, and we are working on on NASH and NAFLD. The first work we are doing is we are working on urine metabolomics in NAFLD and NASH. So this is a spectrum of NAFLD and NASH, where we've started this work, and these are all preliminary data. So what we have taken is we've taken 5 healthy grade 1 NAFLD, grade 2 NAFL, NASH plus cirrhosis, and NASH plus HCC, and these are 5 patients, individual from the groups, and we have taken urine samples and did urinary metabolomics analysis, and we found that we could identify more than 350 metabolites, and of them these red colored dots represent metabolites which was significantly increased in the NAFLD or NASH category when compared to controls. If you see the PLSDA plot, you can see the healthy, which represented in blue color, dark blue color. It is way different to the grade 1 and grade 2 NAFLD as compared to NASH and NASH plus cirrhosis or NASH plus HCC. So NASH plus HCC is completely different, and you can see the NAFLD and NASH are completely different. And this is the VIP scores for the metabolites which are significantly differentially regulated in NASH plus cirrhosis, and this is the VIP score for them. So if you see the heat map analysis for NASH and for the groups, you will see that both the grade 1 and grade 2 NAFLD comes very close to the healthy, and the NASH grade only NASH plus cirrhosis, and NASH plus HCC are way different than what healthy represent, and there are different molecular understanding for this. So what are the biomarkers, what are the protein, what are the metabolites that are significantly regulated? I'm sorry for this blur image, that it says that the metabolites which are significantly different in the NASH category is basically vitamin B metabolism and lipid metabolism, which are significantly increased, and the decreased metabolites are arginine, proline, and nicotinic acid and histidine metabolism. So I have tried to identify certain very important molecules which are significantly different. So these are the 4 molecules which are very, very different, and these are like sphingosine 1-phosphate and I cannot see the name because of so this is from the so I tried to identify, if you see in terms of HCC, the 4-nitrophenyl 3-ketovalentamine is actually a fungal metabolite that was identified in the urine. And if you see, this metabolite is significantly higher in the NASH plus HCC patient. So there may be fungal association of development of HCC in NASH. So this preliminary studies gives us an idea. We also identified 2,4-diaminopentathenate, which comes from the exposomes. So what is an exposome? Is actually whatever the person has acquired or have infection from the starting, it is called as the exposome, and this is from the exposome. Next study which we are doing, and this is also a preliminary study we have just started, is that we are working on urinary metabolomics on treatment of N-acetylcysteine on NASH as well as ASH. So both N-acetylcysteine is very well known to inhibit the hepatic lipid accumulation, oxidative stress, inflammation, and lipolysis. So it is very well known to inhibit this basically, and also known to increase the glutathione, anti-inflammatory property, insulin, and mitochondrial function. So this N-acetylcysteine is routinely given to those patients who are non-alcoholic fatty liver disease or who comes from alcohol. So how does the metabolic phenotype, the urinary metabolic phenotype of NASH differs from ASH? This is what we're trying to understand pre and post NAC treatment. So what we have done is, here we are collecting the patient, and this is just the preliminary analysis again. So we have taken pre-NAC NASH samples N is equals to 8, and the post N is equals to 10, and pre-ASH pre-NAC ASH sample N is equal to 8, and post NAC sample N is equal to 10. We did the urinary metabolomics, and we compare or NASH and ASH for pre and post treatment. The idea is to identify NAC regulated pathway in NASH and NAC regulated pathway in NASH. Are they different, or are they similar, or they behave differently? So these are the differentially regulated metabolites which we identified. More than 350 metabolites were identified based on standard metabolomic criteria, and these are the metabolites which are significantly upregulated. The list of the metabolites that are significantly different, maybe if so you can see that these are the two metabolites, the first one and the second one. I'm not able to see the name because of this is cyclohexanamide and aniline, which are significantly different, and you can see both in ASH and NASH post sample there is significant increase in cyclohexane carboxylic acid as well as aniline in our study. Next, if you see the PLSDA plot, you can see that the NASH and ASH completely differ from each other in terms that the ASH post sample and the NASH post sample, they coincide to each other, whereas the ASH pre-sample and the NASH pre-sample coincide to each other. That means the NAC is having certain changes in the metabolic phenotype of these patients, and these are the changes which we identified. So the most important variable for this projection was 307, and this 307 is basically coenzyme Q2, which was significantly increased. We have significant increase in aniline, and then you have significantly increase in 5-nitrotoluene. So these are the metabolites which could differentiate between ASH and NASH. So if you see the heat map of these metabolites, so you can definitely see that both the post-NASH and the pre-NASH completely comes together, and they are significantly different from each other. In fact, the post-ASH and the post-NASH samples also differ from each other. Finally, the upregulated molecules which are in NASH, the metabolites which are the pathway which are upregulated is thymine metabolism, and the amino acid metabolism, seleno compound metabolism is significantly increased after NASH treatment, after NAC treatment in NASH, and these metabolite pathways were significantly decreased after NAC treatment. These are phenylalanine, tyrosine, and tryptophan biosynthesis is decreased. Phenylalanine metabolism, vitamin B6 metabolism is decreased, and in case of ASH, we found significant increase in glucose-alanine cycle, alanine metabolism, spermidine and spermidine biosynthesis. The glutathione biosynthesis significantly increased, and these like vitamin B6, which was found in the NASH also, they were significantly downregulated. Betaine and tryptophan metabolism, which is significantly downregulated. So as this conference is based on cancer, we have very important data that is associated to cancer, that is gallbladder cancer, and we have analyzed bile. I wanted to show these slides also in terms. So gallbladder carcinoma is actually known to be the 5th most important gastrointestinal malignancy, and the incidence is very, very high in the southern part of the country. So if you have a gallstone, what happens is that the bile actually flows out of your body and comes into the body, and then you can see icterus, or you can have itching, and there are many other complications that happens. So why do you want to identify gallstone? What are the factors? What are the risk factors? Cholestasis are the risk factor. Old age, being a female, is the risk factor. If you have typhoid carrier like chronic infection, IBD, these are the risk factor. And the size of the stone actually determine the cancer, how bad would be the cancer. So if you have a higher chances of cancer, if you have the bigger size of the stone. So why bile? Bile is actually the blood of the gallbladder, and if you analyze bile, so you are more subjected to identify certain the microenvironments associated biomarkers that are associated to cancer. Therefore, bile was chosen for analysis. So what we did in this analysis, we did 2 analyses. We did lipidomic analysis as well as metaproteomic analysis, and I'm going to brief. So in the training cohort, there were 87 patients, and in the test cohort, there were 38 patients with bile as well as plasma samples. So what we did is we did this lipidomic analysis. This is the workflow for lipidomic analysis, and this is the patient profile. If you see that the patient with CAGB was significantly more sick compared to those which were not with gallstone gallstone or healthy control. And if you see that the total bile acid was significantly higher in gallstone patient, CAGB with gallstone as compared to only CAGB, and the cholesterol levels or the lipid levels were significantly decreased in the CAGB patient as compared to gallstone or healthy control. So this was similar for us in the training cohort as well as the test cohort. So if you see the metastability of the bile, which is actually seen. So metastability of the bile depends on the percentage cholesterol, percentage lecithin, and percentage bile acid. And in our study, you can see that the bile was the metastability of the bile was lowest in patients with CAGB. So when we did lipidomics in CAGB and compared it with gallstone and healthy control, we could identify 466 differentially expressed lipids. In CAGB patient, 82 upregulated, 384 downregulated when compared to gallstone, and 490 upregulated and 79 upregulated and 411 downregulated when compared to healthy control. If you see the PLSDA plot, you can see that the CAGB were completely different from the gallstone or healthy control. We did the lipidomic analysis and we identified that most of all the lipids were significantly downregulated, like phosphatidylinositol, phosphatidylethylamine, phosphatidylserine, phosphatidylcholine was significantly downregulated and shared significant correlation with each other. And these are the lipids which were significantly associated. And based on the top 5 lipids, we did the probability of cancer development analysis, and we found that the probability was more than 90% in the patient with CAGB. Now we did the bile metaproteome analysis in CAGB. This is a newer technique by which what we do is we identify bacterial peptides using LCMS, and this gives you better results than 16S. That, for example, if you do 16S, how many phylum you may identify, but in metaproteome you identify more than 17 phylum. So we did identify more than 17 phylum. So these are the phylum expression status for healthy control, gallstone with CAGB, and the species level, more than 50 species were identified. If you see the alpha and beta diversity of the gallstone CAGB with gallstone or CAGB alone, you see that the alpha diversity as well as this was significantly based on the Shannon and Simpson index, as well as the beta diversity was significantly different when compared to gallstone and healthy control. And we did the functional analysis. The best part with metaproteome is that you can do functional analysis, and like in pie crust, this is hypothetical. In here, it is not hypothetical because you have the expression of certain bacterial enzyme which results into the COG function analysis. So you can see that the COG function analysis, there was significantly decrease in lipid transport activity in the CAGB and CAGB gallstone patient. And based on this bacterial peptide, we also did the probability of the cancer development analysis, and we found that the probability was greater than 80% in the CAGB patient group. We also compared patient with cholestasis versus non-cholestasis, and you can see that the amount of bacterial phylums was significantly different. The alpha diversity was significantly higher in the non-cholestasis group, and this is the beta diversity, and these are the bacteria which are specific for the cholestasis. For example, Nocardia, Streptococcus eucenas, and Bacillus was specifically increased in the cholestasis group. Then we did the WMCNA and WPCNA. This is the integration analysis, which is very important. And what we did is we integrated the analysis into a multi-modular correlation network. So how did we do that? Is that we took the lipidome analysis and we did weighted lipidome correlation network analysis, and based on this we could identify modules. So these are the modules. And we also did the modular analysis for the metaproteome, and these are the module details. Then we correlated the lipid modules to the bacterial modules and identified those bacterial modules which are correlated directly to the lipid modules, so that we can identify that how the bacterial peptide correlates or associates or change the lipid profile. And we also correlated the bacterial module, the lipid profile to the bacterial functionality that we have. And we can see that these are the lipid modules which correlated directly with the bacterial functionality. Based on this, we sorted this diagram. We identified that these are the LCA which are already known in cancer and identified from our study. These are the LCA which are not known in cancer but identified from our study, that is Firmicutes, Streptococcus cerevisiae, and Proteobacteria hypno microbacteria, which was significantly increased in CAGB bile samples of CAGB. This results into significant increase of the functional analysis, and these are the functional which are significantly increased, that is signal transduction lipidomics, and it directly correlates with the significant increase in the lipid classes, that is LPE, PIP, and N. And then the bacterial proteobacteria significantly decrease the nucleotide transport, and it inversely correlates with the lipids that are identified. Then we correlated the lipid modules with the clinical factors. So here, these are the lipid modules which are known, and they are correlated to bile acid profile as well as the clinical module. And you can see the red color represent direct correlation and blue color represent indirect correlation, where you can see that the lipid module correlates directly with the bile acid and inversely somewhere with the clinical factors. And this was done for the lipid modules for 4 types of bacterial clusters. So these are the clusteral analysis as well. Finally, sir, we identified the lipid species which were significantly differentiating cancers versus no cancer, gallstone in CAGB versus CAGB alone, and gallstone versus healthy. And we identified the bacterial peptide as well, which was significantly differentiating the same. And these are the fold chain values and p-values. So based on this, we took these 8 lipid molecules and we validated them using LC-MS analysis in the bile validation cohort and the plasma validation cohort. And you will see that these are the AU-ROC analysis for them in the validation cohort as well as the plasma validation cohort. These are the structure. This is the MS-MS analysis for the lipid molecules. And finally, we did the machine learning analysis. So we took all the lipid molecules together and in comparison to each other, and we did the kappa analysis and accuracy analysis, and one can see that all the lipid molecules all together have the highest accuracy and kappa value, and showed the highest accuracy, sensitivity, specificity, and log p value for the bile group as well as for the plasma group. And this is this completes the the identification of biomarkers, and it says that all together these lipid molecules can serve as a molecular targets for detection of CAGB as early as in 30 minutes. So finally, I would like to thank my mentor, Professor Shiv Kumar Sareen, and this is my omics group. And I would thank everyone for being so patient, and I'm ready for any questions. Thank you so much.
Thank you so much, Dr. Singh. This was really elaborative. Okay, so considering the time constraint right now, we will move to our next topic, and which will be delivered by Dr. Shantanu Chaudhary. His topic for today is breast cancer genomics, and Dr. Shantanu is currently a professor at the CSIR of Genomics and Integrative Biology in Delhi. His research interests include the understanding mechanism behind telomerase and telomerase control and telomere function in cancer and neurological disorders, including the neural differentiations and tumor organoid biology. Dr. Shantanu received his PhD degree in chemistry from Indian Institute of Chemical Technology at Hyderabad. He is trained as a molecular biologist in the biochemistry department at the University of Nebraska, USA, and at the Max Planck Institute of Terrestrial Microbiology, Marburg, Germany. In 2012, Dr. Shantanu was awarded the Shanti Swarup Bhatnagar Award in Biological Sciences for his work done in India. Welcome, sir. Over to you.
Thank you so much, Upasana. And thank you very much, Professor Ganguly, for inviting me to this conference. And it's really been a very interesting series of talks, very deep, ingrained, and articulated. What I'm going to do in the next 10 minutes or so is that give a very fairly broad overview, very relatively high level overview of work that we initiated about a year and a half back. And it's so to give to sort of articulate the intricacies of how we are putting the teams together, what are the mechanisms of how we have started working, what are the basic tenets of what we are doing, and also sort of give a flavor of what is very recent emerging data. I would not have time to go into the details, but probably in a future opportunity, I'll be able to share more details on the analysis. So the to this audience, it is today's audience, I can skip the part of which I generally start with, the part of the importance of multiomics in clinical decision making, which is the topic of today's conference also. So, and with this in mind, we initiated this study. As we all know, the Cancer Genome Atlas is very few Indian patients are represented. So there is a gap in our understanding of how do Indian breast cancer typically, and it applies to other cancers also. How do, as a population, how much of this is overlapping with the findings that we have from other populations, and what is distinct and what is similar. To understand this, we put together this program of doing the first 1,000 Indian breast cancer genomes to create an atlas of whole genome sequence, transcriptome, and a few other things, which I'm going to go into in the next few slides. So this is a CSIR, that is Council of Scientific and Industrial Research led program, which is for 1,000 breast cancer, and the broad overarching reason for this is to create a resource for patients, clinicians, researchers to use advances in genomics, which without hard data, one cannot apply all the nuances of genomics that can be applied, requires hard data. So the idea is to create that resource for others to use, and obviously do our own analysis to see how we fare with other samples across the globe. Here is a summary of exactly what we are trying to do. And we are taking treatment naive tissue, and we are just noting what is the treatment that is being given at the hospital, standard of care, and then response to therapy is taken along with clinical annotations. With the treatment naive tissue, we do molecular characterization, including cell-free DNA, molecular characterization in terms of whole genome sequence, transcriptome, and epigenome. And then we would like to create a molecular footprint, which is a very well-known. I don't have to go into the details of why create a molecular footprint of the disease in terms of and correlate it with the clinical annotation. Hopefully, this will allow us to form strong association patterns, discover association patterns, which can be applicable across groups of patients and how they respond to therapy. With that in mind, I think the program started with discussions and conversations with Dr. Sudeep Gupta, as you know, is very experienced in this field. He has given a talk before me in this session, and we know how intricate the data and the studies that were taken up. So Dr. Sudeep Gupta has been has been there with us right from the beginning of the program. He's also a collaborator. And Dr. S.V.S. Dev from All India Institute of Medical Sciences, they were the initial pioneers with whom we discussed and we put together the program. And we have a very illustrious advisory board. Dr. K.S. Gopinath is a Padma Shri and a surgeon from HCG hospitals in Bangalore. Dr. Chitra Sarkar is a very well-known pathologist, senior pathologist working in AIIMS, New Delhi, and a few others. And with their suggestions and with their inputs, we formulated this program. And the previous slide was basically capturing the nuances of all the discussions that we had. The initial partners, and now it has, I'll come in a few slides, we'll talk about the others. The initial program started with obviously Dr. Gupta and his team from Tata Memorial Center, ACTRA, and Dr. Dev and his team from All India Institute of Medical Sciences, Dr. Anurag Mehta and his team from Rajiv Gandhi Cancer Center in Delhi, and Dr. Samir Bhattacharya and his team from Saroj Gupta Cancer Center in Kolkata. And later on, many others have joined. In the last update, we know that we have, other than that, Sir Gangaram Hospital, with Dr. Professor Ganguly's help, who is chairing today's session, we have been able to include now Apollo hospitals in this program, Sri Sankara, then Dharmashila, Majumdar Shaw Medical Foundation, CMC Vellore, or other partners, and there are a few others with whom we are constantly conversing, and hopefully we'll be able to join. On the analysis side or the data collection side, the program is led from my own organization, IGIB, and then we have partners like CSIR CCMB, CSIR IMTech, and CSIR Indian Institute of Chemical Biology. The analysis is primarily done with NIBMG, National Institute of Biomedical Genomics, and then there is supporters of this program are also ICG and Persistent. So here is a top level summary of what we are doing. As I said, NGS, DNA, RNA, and protein, definitely we are doing that. And then we are also making, for the first time, I think we are making organoids from patients. We are from the patients for which the genomic characterization has been done. We are making organoids of this, and we are asking people to take these organoids from us for their own research, where we're giving all the annotations of the organoids along with the genomic and transcriptomic characteristics, and asking them to ask their biological questions. Ourselves, we are putting them through a drug screen. And a liquid biopsy is also, or cell-free DNA analysis is also a part of this, with the idea of to see whether how much of this correlates. As Dr. Sudip Gupta showed, the ctDNA or the cell-free DNA is a very strong correlate of what is happening at a molecular level. So a little bit deeper idea of the program. So we take both prospective and retrospective samples and collect tissue, and we take normal, adjacent normal, wherever the surgeon is able to tell us that confidently that this is surgically removed normal, and wherever it is possible. And otherwise we use blood as for germline correction. And also we collect blood at every follow-up that patient comes for. We collect blood, and hopefully 10 ml of blood for doing cell-free DNA to sort of see how the patient is responding. And we would eventually, after three years of follow-up, we would have the clinical response of the person or patient, and this would be correlated with the molecular analysis. So this is the summary of the previous slide in some sort. We ask for about 200 mg of resected tissue or 80 mg from each core biopsies, 2 to 3 cores. Sometimes it is possible, sometimes it is not. And we work with even lesser samples. We have been able to do things and confirm that more than 60% tumor tissue, first draw before surgery, no medication given. This is very important for us because we are saying that these are all treatment naive at presentation. And then follow up patient for 3 years. So 200 milligram of tumor and 200 milligram of adjacent. We ask this to be at in a RNA later, and this RNA later shipped to us, and the blood sample happens to be in a different protocol for that. This is the logistics of how the program is being done, because as you could see, it's a multi-center program spread across India, and we hope to engage more partners across India. So it is very important for us to make sure that the logistics of the program of transport is also in very well articulated. This is what we envisage to get, and these are not new to this audience. Disease subtyping and classification, prediction of biomarkers, of course, derive insights into the disease biology. That is why we have the organoid biology program going on along with this, and also understanding complexity of cancer host. And so that part of my lab is interested in the immune component of cancers. So we work on, particularly with TNBC, we work on the immune host immune interactions. So very briefly, the progress so far. We have collected about 300 breast cancer patient samples, and more than 250 of these have been sequenced, both normal and tumor. And the median depth of sequencing is fairly deep, which is about close to 65 to 70x of whole genome sequencing. And then there is transcriptome at about 60 to 80 million reads. And of course, we have the clinical staging treatment and the follow-up data from the hospitals. So far, the collected samples are about half of them are triple negative breast cancers, because we initially focused on triple negative to build a core for triple negative, and then followed up with more with non-TNBC. So as we go ahead, this will more or less represent what is seen across India in terms of percentage, and it is roughly going to be around 25 to 30% TNBC from what I can see from the samples that we have collected so far. This is how the samples that we have received, and we see that this is the age group distribution, and this is also the receptor distribution in terms of and the age distribution of the receptor, more or less what is seen across the world. A little probably on the but we'll need more data to support that. A little lower on the age side in some times, particularly for triple negative, but that's very emerging data right now, and hopefully we will be able to confirm that with more samples. This is a summary of an analysis, that is the somatic analysis that we have done, and we were able to see some of the genes that have already been discussed, and then there were some others which were not being discussed so much, but we see them in a few clusters. We do see them. Some more intricate analysis is ongoing, and hopefully, as I said, we'll have a future opportunity to discuss them in much more detail, including some interesting amplifications and deletions that we see now in patients, which are somewhat different from what has been noted across other studies done so far across the world. And those are very interesting things. We are trying to validate and confirm that we are on solid ground there. This is a comparison of TNBC across 158 TCGA dataset published in 2015. Then there was a China study published a few years back. And this is the age distribution. What we see in the first 64 triple negative patients that we had, we saw age distribution as it's reflected here, more or less similar with little bit nuanced issues that we can, once you zoom in and you can see some interesting differences. And Indian versus TCGA stage comparison also matches more or less. Few changes are there, and we are eagerly waiting for more number of samples to sort of understand that this is robust and does reflect a little bit difference and from what has been noted earlier. This is just a snapshot of the differential analysis that we have done so far, and there are several interesting things which I don't have time today to point out, but just to say that we have been doing this very aggressively so far. We released this data set for public use of everybody. 200 plus samples were released from our a few days back. And this is how the website looks, and one is able to see that we have 200 plus tumor normal whole genome sequence at a fairly high depth of close to median 70x coverage. And as of now, there's more than 450 clinicians involved across 12 different hospitals in India, more than 35 scientists involved in the different isolation, sample prep, then also in terms of analysis, sequencing and analysis, 12 hospitals and institutions. Some of the major partners so far are highlighted here. Others are also joining as we speak. This is how the website looks. One can filter and choose what kind of patients in case you are interested in a particular type or a particular class or a particular receptor status. One should be able to do that very fairly easily. And then you choose this selections, and then you can have some idea of what is the clinical status. And once this is available for download, after a request is made, and this request is verified by a data access committee to make sure that this is a genuine user and the use is also for a certain purpose. And once you do that, you should be able to freely download this data and work with this and do your own analysis. And hopefully this data set will also grow as TCGA like TCGA at some point, and be able to make some robust give some robust help to clinicians across the world or researchers across the world to compare Indian data along with others. This is where the database is accessible from, ibcga.igib.res.in, and please go ahead and put in a request if you are interested. We will take it to our data access committee and get back to you in a couple of weeks to opening up. And with that, I would like to thank you. There are numerous people, and I have sort of named the hospitals and the institutions. In each institute, we have 5-6 people, or at least sometimes more than that, in a team across hospitals. I'm not taking, but every one of them have contributed so far very eagerly into this, and also very strong regulatory processes we take care of, so that there are all understandings for clinical data, ethics approvals, consent are taken care of and kept in. So I will stop there with this. Thank you very much.
Thank you, Dr. Chaudhary. Thank you so much for this elaborative talk. Now I would request Dr. Ganguly to join in and start the question answer session. I don't see much questions on the chat box. It is open to the audience. If you have anything, please ask right now. Our speakers will be happy to address them. There are 2 questions to Dr. Sudeep, which has been already answered by him in the chat box.
So Vasana, because for the sake of the time,
Yes,
please address any question to any one of the speakers, and they will be glad to provide the answer. As well as if you want to collaborate, you could also collaborate with any one of them. What today we saw in Shantanu's presentation, that how many of the scientists with the diverse institutes, diverse background could come together to create a atlas which could be used by almost everyone. So majority of the data which he is doing will be an open source data, and he also offered that organoids, which is a very good system to ultimately move towards in-depth analysis of particular phenotypes and genotypes, will also be available to people. So this is a very good news, and we could really hope to utilize all these things. And in Sudeep's paper, we saw that how well we could use the genomic data, even the RNA-seq data, single cell data, founder gene data which are mutating and what's happening over there. Very nice use of all those, and ultimately the clinical protocol which are drawn out of that is lidocaine data was absolutely marvelous. This helps you to stop metastasis, and theranostic tools are also was very, very obvious. The Jaswinder's data was, I will look at few major pointers. The one pointer was this, that how the genesis of the fibrosis and later on pointed towards the hepatocellular carcinoma was unraveled with a very complex metabolic and proteome pathways. And the lipidome was used so excellently for the bile acid analysis, as well as in the pathogenesis of the gallbladder cancer. But what came out as a very major important tool was the tools in which you, without doing a microbiome analysis, how you could really look at the bacterial signatures which control the lipid pathways and other metabolic pathways, and thereby contributed in the overall cancer system. We in Rashmi's paper, we saw that a very dangerous cancer, where even before the signatures in the PET scan and MRI has come up, in a simple blood sample, you could identify the grade and how it is moving. She also found out that this is a target. She is doing a small molecule analysis for these targets. She also observed that developed some simple diagnostic tests out of that, and ultimately she is now doing a work for which she didn't provide those data over there, that how the CRISPR-Cas, she has identified 25 gene signatures, and how the CRISPR-Cas could actually nullify few of them and turn the tide of the glioma, which is a very dangerous disease. Also, she is developing because you have to cross blood-brain barrier. She is using exosomes and some of the drugs as well as the other interventions through spions. So this is a very landmark study in glioma. And in the cancer, we showed you that how lung cancer, we showed you that how the multi-omic data has ultimately resulted in interventions. And one thing which comes out of all these studies that there is a central dogma in the cancers. And these central dogma, they go almost for any cancer, and the how the cancer changes your immune system, and how the immune system could be trained to attack cancer, how the dense matrix of the cancer could be penetrated by the secretomes and others, and how the metabolic pathways could be turned into conquering the cancer by various other means, and how the microbiomes we have seen throughout that how they could be a decisive factor in helping the cancer treatment to move on. We will see that with these, there are a lot of data which is emerging on microRNA in the transcriptomes and other data sets, which will ultimately lead into designing different kinds of protocol. I visited Mofat, the biobank. We have some very good biobanks in India. The one is available in the THSTI, another one is available in the ILBS, another one is available with the Apollo in the Sapiens, and Tata Memorial Center. So there's several, and also in the NIMHANS, the Neurologic Biobank. So these biobanks will be very important in doing this. And in Mofat, I saw that 265 clinical protocols emerged from these omics studies. So this is the future, and very happy to see that how the clinicians are using this data to various places, and these are not actually depending on these sequencing shops. Their own data is emerging, which is much more powerful. With this, I will thank Upasana, I will thank Dr. Nandkumar Jairam, I will thank Sudhir Bhai, Anju Agarwal, and many others who have joined, and Dr. Anupam Sibbal, who is one of the very major force around this, and all my speakers today. They had been absolutely wonderful, gave cutting edge science, and we could finish almost on time, few of 10 minutes overshot. But with this kind of thing, there's a very minimal overshoot. So back to Upasana Mukherjee, and you could now close the symposium.
Thank you so much, sir. Thank you. I would like to personally thank all our speakers, esteemed speakers, Dr. Rashmi, Dr. Jasvinder, Dr. Sudeep, Dr. Shantanu, and none other than Professor Ganguly. Thank you so much for making this time, and this all the topics were very much insightful. I hope people, scientists like us, are very much inspired right now to work more and more on the field of omics, and we really look forward to the great data curation, what is happening in India right now, which is very much important to understand our own population. Thank you so much. Thanks to all our kind audiences who have got time and made time to join us here. Thanks to the organizing team and Dr. Sibbal and all the entire team of the Apollo and the GAPU. Thank you so much.
Thank you. Goodbye. Have a nice dinner.
Thank you, sir. Thank you very much. Good night,
Thank
sir.
you so much.
And in
Good night.
United States, have a nice breakfast. And in Europe, have a nice lunch.
Thank you, sir.
Thank
Bye
Good
bye.
night, sir. Bye.
Bye. And very grateful that all of you
Thank you, sir.
came. Thank you. Bye.
Thank you. Good night.
Dr. Sim.